You know, when you imagine wandering through a dense, quiet forest, foraging for wild food, there's usually this very specific, comforting aesthetic that comes to mind.
Right, like a very curated, almost pastoral illusion.
Exactly. You picture like pastoral innocence, the bright red of a wild berry, or maybe the delicate edges of a golden chanterelle catching the sunlight. It feels idyllic. It feels gentle.
But nature isn't really a curated garden.
Yeah, definitely not. And occasionally you step off the trail, push aside some damp autumn leaves, and you stumble across something that just violently shatters that entire illusion.
Oh, yeah. Like the organism we're looking at today.
Right. Imagine looking down at the soil, and instead of a normal plant or a standard mushroom, you see something that looks like, well, a piece of melted black wax, just dropped right there on the forest floor.
It's an incredibly bizarre visual. It's shaped like a distorted alien saddle.
Yeah, an alien saddle. And if you lean in closer, you notice the stem isn't smooth at all. It's deeply grooved, hollowed out, and heavily fluted. And, you know, if you pick it up, you might find that this weird, waxy structure is actually housing a bustling ecosystem.
A literal multi-level apartment complex of millipedes and beetles hiding in those crevices.
It's crazy, but the visual is really just the beginning, right? Because if you take that strange saddle into a lab and dismantle its cellular structure, you don't just find standard organic compounds.
No, you absolutely do not. Hidden deep inside its tissues is the exact literal chemical compound used to fuel the orbital maneuvering engines of the space shuttle.
Which is just mind-blowing. So welcome everyone to a deep dive into the organism known to science as Helvella lacunosa.
Commonly known to foragers as the Elfin Saddle.
Yes, the Elfin Saddle. And our mission today is to completely deconstruct what biologists call the Cosmopolitan myth surrounding this fungus.
We're going to get into how modern DNA sequencing completely upended centuries of botanical history.
Exactly. We'll show you how this isn't just a single mushroom, but actually a massive hidden network of dozens of distinct species operating in plain sight.
And we're going to decode its brilliant biomechanical engineering, too.
Right. And most importantly, we're unpacking its incredibly complex dual nature chemistry, a profile that spans from deadly, volatile rocket propellant to molecules that are actively being researched as revolutionary cholesterol-lowering medicines.
It's a huge spectrum, and to map out a profile this complex, we are synthesizing a really diverse array of source material today for you.
Yeah, we've got cutting-edge phylogenetic research papers tracing the evolutionary genetics from places like the Chinese Academy of Sciences.
Along with work from mycologists in the Pacific Northwest, clinical toxicology reports, and ethnomycological studies from Mexico.
Plus, you know, practical field foraging guides to ground it all. And honestly, the best way I can conceptualize the Elfin Saddle after reading all this is to imagine a highly trained international spy.
Oh, I like that analogy.
Right? Like a spy operating with dozens of different passports. Because this mushroom looks utterly identical everywhere it goes. It wears the exact same deeply wrinkled, dark gray disguise, whether it's sitting in a forest in Sweden or hiding in California pine needles or on a rotting log in Tibet.
But its true identity, its actual genetic code changes entirely depending on what continent or even what specific forest it happens to be operating in.
And for over 200 years, that visual disguise worked flawlessly against the brightest minds in biology.
It really did. The story formally kicks off in 1783 with a Swedish naturalist named Adam of Zelius.
Right, of Zelius. He's the one who documented it and officially named it Helvella lacunosa.
Exactly. And for centuries after him, the global scientific community operated under the strict rules of morphological identification.
Which basically means identifying an organism just by its physical shapes and characteristics.
Yeah.
Right?
Yeah, precisely. Scientists looked at the shape of the cap, the color of the tissue, the fluting on the stem, even the microscopic size of the spores. And because all those physical traits matched perfectly across the globe, the textbooks definitively stated that this was a single, widespread, cosmopolitan species.
The cosmopolitan myth, I mean, it's a very human assumption to make. We like the idea that nature is uniform.
We want things to be neat and predictable.
Right. If you see a black, waxy, fluted mushroom in England and the exact same black, waxy, fluted mushroom in California, it feels logical to assume they're the same thing.
But then the molecular revolution comes along and completely rewrites the rule book.
Thank goodness for DNA sequencing.
Yeah, it was akin to turning on the lights in a very dark room. Modern researchers started looking at specific genetic markers, what they often call fungal barcodes.
Okay, fungal barcodes, like scanning an item at the grocery store.
Kind of. Think of the mushroom's genome as a massive encyclopedia. If you want to figure out how two organisms are related, you don't read the whole thing. You just check specific, highly conserved chapters.
Regions of DNA that govern basic cellular functions. And because those chapters are so critical for survival, they don't tolerate random mutations very well, right?
Exactly. They mutate at a very slow, measurable rate. They act like biological clocks.
The sources mention regions with complex acronyms, like the internal transcribe spacer, the ITS, or genes like NRLSU, TEF1A, RPB2, and HSP90.
Right, those are the specific barcodes. By comparing the microscopic spelling errors in these regions across different Elfin Saddle specimens, scientists can calculate exactly how long ago these mushrooms shared a common ancestor.
And when they finally ran those barcodes for Helvella lacunosa, well, the entire Cosmopolitan species concept just collapsed.
Completely fell apart. It is absolutely not one globally roaming species. It's what we call a species complex.
A complex consisting of at least 49 distinctly different genetic lineages scattered across the world. 49.
It's a massive number. That means the original mushroom Atomacilius found in Sweden back in 1783, the one scientists now call the true lacunosa in the strict sense, is just one tiny fraction of the whole picture.
Yeah, the data shows that specific original species is actually mostly restricted to Europe and certain pockets of eastern North America.
And once you cross the Rocky Mountains and move into western North America, the genetics diverge entirely.
Even though they still look exactly like that melted wax saddle.
Right. Millions of years of geographical isolation and unique ecological pressures force those western populations down completely different evolutionary paths.
For example, the sources talk about a species in the West called Helvella vespertina.
Yes, vespertina. It tends to grow slightly larger, more robust, and it's formed deep ecological alliances primarily with coniferous trees.
Your Douglas firs, your Monterey pines.
Exactly, and its biological clock is tuned to the darker, colder months. It typically fruits between October and January.
But then, in the same broad geographic region, you have another completely distinct species, Helvella dryophila.
Right, Dryophila. It favors a squatter, almost pure black cap, often with a pale stem when it's young.
And it completely ignores the pine trees. It prefers oak trees. Plus, it fruits later in the winter and spring, roughly January through June.
And that's just North America. The complexity scales up dramatically when researchers turn their sequencers toward Asia.
The data out of the Chinese Academy of Sciences is just staggering. They identified 25 unique genetic lineages within China alone.
And out of those 25, 16 were completely novel species that science had never documented before.
Incredible. You have Helvella australo-occidentalis, which specifically adapted to digest rotting ancient tree trunks in high-altitude Tibetan environments.
And in stark contrast to that, you have Helvella huangi, which remarkably adapted to thrive in heavily modified, concrete-laden urban soils right in the heart of Beijing.
So we have 49 different species, separated by oceans, separated by millions of years of evolution, adapted to completely different trees and climates.
But to the naked eye, they all look identical.
How does that even happen? If their DNA is mutating and diverging so radically, why wouldn't their physical appearance change to match?
The biological mechanism driving this is called cryptic speciation. It happens when organisms undergo deep genetic divergence. They become separate species and can't interbreed, but their physical morphology remains static.
And the main driver for that is something called stabilizing selection, right?
Exactly, stabilizing selection.
Let's break that down for the listener. Are you saying the environment is essentially locking their physical shape in place?
That's a great way to visualize it. Imagine you design a perfectly aerodynamic car chassis. It cuts through the wind, handles the terrain flawlessly. Over the years, you might upgrade the engine, swap out the internal electronics.
The internal mechanics, the genetics, are entirely different.
But you never change the external chassis because the physical environment it operates in hasn't changed.
Wow, okay. So for the Elfin Saddle, the physical demands, pushing through leaf litter, dispersing spores, surviving autumn rains, those are relatively constant across these global habitats.
Right. Evolution is incredibly pragmatic. If a physical design solves the environmental problem perfectly, there is zero evolutionary pressure to change it, regardless of what the underlying DNA is doing.
That makes total sense from an evolutionary standpoint.
Yeah.
Brilliant survival strategy. But I have to push back a bit here on behalf of the listener.
Go ahead.
is an absolute nightmare for anyone actually trying to interact with this organism in the real world. Think about it. You're walking in the woods, carrying a field guide. You don't have a DNA sequencer in your backpack.
No, you certainly don't.
You can't just casually run a PCR test on a Sunday hike to see if you're looking at Vespertina or Dryophila. If the visual disguise is literally perfect, how are we supposed to know what we're dealing with?
It is a massive source of frustration in the foraging and mycological community right now. The shift from morphological to genetic identification is revolutionary, but practically very difficult in the field.
So what's the solution?
The solution requires a fundamental shift in how we observe nature. You have to stop looking solely at the object itself and start deeply analyzing the context surrounding it.
Like profiling the spy based on their environment, not their face.
Exactly. When dealing with cryptic species complexes, the environment provides the identifying markers the mushroom refuses to show. You have to become an ecological detective.
So you look around, are you standing under broadleaf oaks or Douglas firs?
Exactly. What's the ambient temperature? Is it the freezing dark of December or the warming soil of late April? The host tree and the specific microseason become your only reliable clues.
That's fascinating. And it makes me think, if evolution decided this specific, weird, waxy saddle shape was so flawless that it preserved it across 49 species, we really need to look at the mechanics of that shape.
We do. There is profound functional genius behind looking like a melted piece of wax on a fluted pillar.
Let's get into the biomechanical architecture of the Elfin Saddle. It really is a masterclass in biological engineering, starting with the cap, the pileus.
The pileus generally measures anywhere from 2 to 10 centimeters across, and it defies standard mushroom symmetry. It's deeply wrinkled, irregularly lobed.
Almost tacitly pinched or folded. And the coloration is so subdued, it absorbs light instead of reflecting it, just matte black or slate gray.
And the top surface of that waxy cap is the hymenium, the fertile spore-producing tissue.
Which is so weird because when most people think of a mushroom, they picture the white button mushroom from the store. You flip it over, and underneath the cap you see gills.
Or a porcini with its sponge-like pores.
Right, but the Elfin Saddle has none of that.
And that structural difference comes down to deep evolutionary divisions. The gilled mushrooms belong to a group called Basidiomycota, which passively drop their spores into the wind. But Helvella belongs to a completely different phylum called Ascomycota.
They don't rely on gravity at all.
Oh.
The fertile surface of their cap is packed with microscopic pressurized structures called
Yes. Let's visualize an ascus. It solves a critical aerodynamic problem. Right next to the ground, there is a boundary layer of dead, stagnant air. If the mushroom just gently dropped its spores, they'd get trapped in that stagnant zone and fall straight to the girt.
Oh, I see. So by using high-pressure hydraulic launching, it blasts the spores through that dead air and up into the active wind currents.
Ensuring they can disperse over vast distances.
The cap is essentially an actively firing battery of microscopic water cannons. That is wild.
But that cap is heavy, and it needs support.
Which brings us to the stem, the stipe. It stands about 3 to 15 centimeters tall. And unlike a normal, smooth mushroom stem, this one is heavily textured with deep, cavernous longitudinal grooves.
Those specific grooves are known botanically as lacunae. It's the defining physical characteristic that gave it the name lacunosa.
But the true structural genius is hidden inside. If you slice that stem cleanly in half, it is not a solid column of fungal tissue.
No, it's entirely hollowed out, constructed from a complex network of internal chambers, supported by thin, interconnecting fungal walls.
When I was reading about this, my mind immediately went to Gothic architecture, like the Notre Dame Cathedral. The medieval architects didn't hold up that heavy roof with solid, impenetrable stone walls.
They used flying buttresses, skeletal, vaulted, fluted columns.
Right, supporting immense vertical loads while using a fraction of the stone. And the Elfin Saddle is executing the exact same principle of structural engineering millions of years before humans figured it out.
The physics translate perfectly. Creating biological tissue requires a ton of metabolic energy, water, and nutrients. A thick, solid stem would be a huge waste of resources.
Just to support its own weight.
Exactly. By using a fluted, chambered buttress design, the organism gets maximum vertical rigidity and structural integrity with minimal biomass.
And it needs that flexibility. These mushrooms fruit in late autumn and deep winter. That hollow column has to endure getting pounded by heavy freezing rains and bend under the weight of winter snowdrifts without snapping.
It's incredibly resilient, but There's an interesting ecological side effect to building a hollow architecture full of crevices.
Right, because in nature, empty space never stays empty for long.
Exactly.
Yeah.
A physical structure unwittingly creates a highly specialized microhabitat.
The foraging guides call it the millipede mansion.
I love that name.
Me too. Because if you pluck one from the soil and look closely at those deep lacunae, it is invariably teeming with life.
The deep grooves provide perfect refuge. For small invertebrates, millipedes, springtails, beetles, fungal gnats, the forest floor is a dangerous place.
Flooding, predators, sudden temperature drops.
Right. But the intricate architecture of the Elfin Saddle provides a sheltered, temperature-stable environment.
Which is great for the bugs, but a logistical nightmare for foragers. You can't just wipe it off with a towel. You have to meticulously use running water and tiny brushes to actively evict an entire apartment complex of insects before you can take it to the kitchen.
It perfectly illustrates how one organism becomes foundational architecture for a whole microcommunity. The stem acts as a protective fortress on the outside.
But, and this is where our deep dive takes a very dark, sudden turn. While the physical structure is a sanctuary, the chemical structure operating inside the cells is highly lethal.
The toxicity of this mushroom is truly something else.
We have to talk about the rocket fuel.
Yes.
When I first read the toxicology reports, I genuinely thought I was misreading the data. It sounds like science fiction, but it is a hard, verified biochemical fact.
It's one of the most astonishing examples of chemical extremity in the natural world. The cellular tissue of the Helvella lacunosa complex is saturated with a highly volatile mycotoxin called gyromitrin.
Gyromitrin, a relatively small molecule, carbon, hydrogen, nitrogen, oxygen. And if it just stayed intact, it might not be a catastrophe. But human stomachs are basically vats of strong hydrochloric acid.
Exactly. When a person consumes raw or undercooked Elfin Saddle, the gyromitrin hits that stomach acid and a violent chemical reaction begins.
The acid triggers hydrolysis.
Right. Water molecules in the stomach acid cleave the chemical bonds of the gyromitrin. It degrades into an intermediate, unstable compound called N-methyl-N-formylhydrazine, or MFH.
But the reaction keeps going.
It does. The MFH metabolizes further, stripping away molecules until it synthesizes a final compound, monomethylhydrazine, or MMH.
And let's be very clear for the listener. When we say volatile chemistry, we usually mean it metaphorically. But here, it's literal. Monomethylhydrazine is the precise, literal liquid chemical propellant utilized by NASA.
The exact fuel that powered the orbital maneuvering engines of the space shuttle.
It's used in satellite propulsion systems. It is violently reactive, highly unstable, and extraordinarily toxic to carbon-based life forms.
And synthesizing rocket fuel in your stomach is a massive problem because MMH is an insidious, highly specific neurotoxin. It directly targets and dismantles your central nervous system.
Walk us through that mechanism. Once the MMH enters the bloodstream, what is it looking for?
It specifically targets vitamin B6, particularly the active form, pyridoxal 5-phosphate. MMH is highly reactive with B6. When they collide, the MMH binds to it, locking it up and forming a useless hydrazone compound.
And vitamin B6 isn't just a passive nutrient. The brain absolutely relies on it as a biological key for an enzyme called glutamic acid decarboxylase.
That specific enzyme is a factory, and its only job is to manufacture GABA gamma-aminobutyric acid, the primary inhibitory neurotransmitter in the human brain.
To use a mechanical analogy, if your excitatory neurotransmitters are the gas pedal, causing neurons to fire, GABA is the brake pedal.
Exactly. It constantly regulates the system, calming electrical activity so neurons don't fire out of control.
So let's trace the domino effect. The rocket fuel binds to the vitamin B6. The B6 is locked up, so the enzyme factory shuts down. Factory shuts down, so the brain runs out of GABA.
The chemical brakes are completely severed.
The brain is now pressing the gas pedal to the floor with no way to stop.
And the resulting neurological crisis is profound. But it's delayed. There is a latency period while the gyromitrin hydrolyzes and the GABA reserves slowly drain.
It takes anywhere from 5 to 12 hours after eating the mushroom to hit critical mass, which is terrifying. You could eat a meal, feel fine, go to sleep, and the rocket fuel is just quietly brewing in your gut.
When the GABA finally runs out, the initial onset is severe gastrointestinal distress, nausea, vomiting, coupled with profound dizziness and heavy lethargy.
And as the nervous system loses its inhibitory control, the electrical storms in the brain escalate.
Patients experience severe muscle tremors, complete loss of motor coordination. In advanced stages, unchecked neurological firing leads to uncontrollable, violent seizures and eventually coma.
Not to mention the liver and kidneys undergoing acute, catastrophic injury trying to filter it out.
It's incredibly severe.
And the danger isn't even limited to ingestion. The sources detail something the medical literature darkly refers to as the roommate poisoning effect.
Yes. Because monomethylhydrazine is highly volatile, its chemical bonds are weak enough that it transitions from a liquid in the mushroom tissue into an invisible aerosolized gas very easily, especially when heated.
So if someone brings Elfin Saddles into a poorly ventilated kitchen and starts boiling or drying them, the thermal energy causes the internal MMH to vaporize.
Steam rising off the boiling pot is literally laced with aerosolized rocket propellant.
Meaning a roommate sitting at the kitchen table, just breathing the air, can inhale the hydrazine gas directly into their lungs. It bypasses the stomach entirely, goes into the bloodstream, and initiates the exact same central nervous system toxicity.
The same GABA depletion, the same seizures.
Just from breathing the steam.
And researchers are uncovering a much longer-term, darker implication. Clinical toxicologists are scrutinizing a potential link between chronic low-dose ingestion of these hydrazine-containing fungi and ALS amyotrophic lateral sclerosis.
Lou Gehrig's disease. And the evidence for this comes from a really haunting study conducted in the French Alps, right?
Yes. Researchers noticed a statistical anomaly, a geographic hotspot of ALS cases in a specific mountainous region. When they investigated, they found a strong correlation between the people developing ALS and the traditional seasonal consumption of false morels,
Which belong to the same family and use the exact same gyromitrin chemistry as the Elfin Saddle.
But the mystery was that not everyone who ate them developed the disease. Toxicologists had to look at the victims' genetic metabolic profiles. They discovered the vulnerability comes down to a process in the liver called acetylation.
Acetylation, that's essentially the liver's chemical disposal system, right?
Right.
Attaching a molecule to a toxin so you can safely pee it out.
Exactly. Humans are roughly divided into fast acetylators and slow acetylators. Fast acetylators have highly efficient livers that process trace hydrazine and flush it rapidly.
But slow acetylators have sluggish enzymes. They can't clear the monomethylhydrazine fast enough.
So the toxin lingers and accumulates. The hypothesis is that for slow acetylators, years of eating seemingly safe, traditionally prepared mushrooms leaves microscopic amounts of unvaporized rocket fuel in the system,
Causing slow, irreversible oxidative damage to the motor neurons over decades, manifesting as ALS.
It's a tragic progression.
When you lay all that out, the violent stomach hydrolysis, the GABA depletion, the aerosolized vapor, the motor neuron damage, I have to step back and ask a fundamental question. Why?
Why does it produce this?
Exactly. Nature doesn't build complex chemical factories by accident. Why would a forest fungus evolve to contain the exact properties of space shuttle fuel? Is it actively targeting mammalian nervous systems?
It's a brilliant question. While it's devastating to a mammalian brain, humans and deer are really just collateral damage. The primary target is microscopic. Hydrazines are incredibly potent, broad-spectrum antimicrobial and antifungal agents.
So it's chemical warfare against its immediate neighbors.
Exactly. The forest soil is a hyper-competitive war zone. Bacteria, nematodes, competing fungi, they'd all happily dissolve the Helvella's tissue before it could mature. By saturating its tissue with gyromitrin, the mushroom creates a toxic, invisible halo.
A chemical grenade that sterilizes the environment.
The fact that this specific hydrazine perfectly fits into a mammalian GABA enzyme lock, destroying the nervous system of anything larger that takes a bite, is just a horrific evolutionary bonus.
Defense mechanism operating on every conceivable scale. But, nature is rarely monolithic. The chemistry of life is nuanced.
It is.
And this is where the narrative pivots so sharply, it's almost hard to reconcile. Because this exact same fungal organism, the one that manufactures neurotoxic aerosolized rocket fuel, also possesses the genetic code to produce compounds that could fundamentally revolutionize human cardiovascular medicine.
The duality of fungal chemistry is one of the most compelling frontiers in modern science. These organisms are master chemists, but to access those beneficial compounds, you have to meticulously bypass the toxic defense grid.
Right. Given everything we just discussed, the idea of eating this is terrifying. But people do. And to do it safely, the traditional culinary preparation is unforgivingly strict.
You cannot just saute it in butter.
Absolutely not. You must parboil the mushrooms in a massive pot of water at a rolling boil for a minimum of 3 to 5 minutes, in a highly ventilated area, preferably outdoors, on a camping stove to avoid the vapor.
And you must discard that boiling water down the drain because it is now saturated with water-soluble rocket fuel.
Many foragers even say to cut off and discard those beautiful fluted stems entirely, because the hollow chambers trap hidden pockets of toxin that the boiling water fails to reach.
It is a high-stakes culinary procedure, but if you execute it flawlessly, you strip away the gyromitrin and you are left with the mushroom's secondary metabolites.
And the recent pharmaceutical research into these specific metabolites is yielding astonishing data. The Helvella lacunosa complex is rich in highly bioactive polysaccharides, often abbreviated as HLP.
Let's define polysaccharides for the listener. We're essentially talking about complex, heavily branched chains of carbohydrates. In this case, primarily composed of specific sugars known as mannose, rhamnose, and glucose.
And scientists wanted to know what these complex mushroom sugars would do inside a mammalian metabolism. So they set up a study using laboratory mice.
Specifically, hyperlipidemic mice. Mice with artificially induced, dangerously high levels of fat and cholesterol in their bloodstream. They gave them an oral dose of purified HLP, about 60 milligrams per kilogram of body weight.
And the metabolic response was rapid and significant. The HLP drove down total cholesterol, plummeted triglycerides, and drastically reduced LDL.
The bad artery-clogging cholesterol.
But it wasn't just a blunt force reduction. While it destroyed the bad cholesterol, the HLP simultaneously caused a significant increase in HDL, the good cholesterol. It actively, intelligently rebalanced the entire lipid profile.
And how does a complex sugar from a mushroom manage that precise metabolic control? It achieves this by operating directly at the genetic level inside the liver cells.
The polysaccharides act as biological signaling molecules. They interact with specific transcription factors, genetic on-off switches. Specifically, the genes known as PPAR-alpha and CPT1-alpha.
And those genes dictate the fundamental rules of fatty acid oxidation. When activated, they instruct the liver to stop hoarding fat and immediately begin breaking it down and burning it for energy.
The HLP is literally reprogramming the mammalian liver's lipid management software at the DNA level.
It's an unbelievable biological mechanism. We're looking at the foundation for an entirely new class of cholesterol-regulating drugs, derived from the exact same tissue that uses rocket fuel to melt a brain's inhibitory pathways.
And the pharmaceutical potential doesn't even stop there. The Elfin Saddle is densely packed with a compound called ergosterol.
Ergosterol is fascinating. It's a vital structural sterol in fungal cell membranes, keeping them fluid yet stable, similar to what cholesterol does in animal cells. But it has a unique, highly reactive physical property. It is a direct precursor to vitamin D2.
So when the raw mushroom tissue is exposed to ambient ultraviolet light, just standard sunlight, the UV radiation physically impacts the ergosterol molecules.
It triggers photosommerization.
The energy from the light literally breaks and rearranges the chemical bonds, transforming it into pre-vitamin D2. The ambient heat then turns that into active bioavailable vitamin D2.
This specific photochemical reaction is incredibly important, as vitamin D is notoriously difficult to source from non-animal dietary avenues. Exposing these wild fungi to sunlight transforms them into a powerful, sustainable, vegan source of a critical nutrient.
And the biotech industry is taking note. They aren't just relying on foragers in the woods. They take the mycelium, the underground network of the fungus, and grow it in massive industrial fermentation vats.
And through a process called directed biosynthesis, they actively manipulate the fermentation broth. By introducing environmental stressors like trace zinc and magnesium salts, the fungal metabolism shifts into defensive overdrive.
Reacting to the salts by dramatically upregulating production of beneficial indole compounds, like melatonin, the exact hormone that regulates human sleep cycles.
The chemical flexibility is staggering.
It really is. It reinforces that concept from the Renaissance physician Paracelsus. The dose makes the poison. But looking at the Elfin Saddle, I think it's more like the preparation makes the medicine.
Oh, that's a good way to put it.
The dynamic is identical to preparing fugu, the Japanese pufferfish. The chef has to perfectly navigate around the lethal tetrodotoxin in the liver to serve the safe meat.
The fugu analogy is highly accurate. It requires deep, respectful understanding of the organism's boundaries.
And this incredible chemical flexibility, lethal neurotoxins alongside complex metabolic regulators, is exactly what allows the Elfin Saddle to conquer vastly different, even heavily disturbed ecosystems.
Which perfectly transitions us to the ecology of this fungus. Because the Elfin Saddle is not confined to pristine wilderness, it uses two completely different survival strategies. It operates as a highly efficient saprotroph, and it moonlights as a complex ectomycorrhizal partner.
Let's define those roles, because they require vastly different biological machinery. When acting as a saprotroph, The fungus is the forest's cleanup crew.
Right. It secretes specialized enzymes into dead organic matter-fallen oak leaves, dead wood breaking down tough cellular walls, and recycling carbon and nitrogen back into the topsoil. It's a lone wolf digesting the dead.
But when it switches to an ectomycorrhizal role, it engages in an incredibly complex symbiotic trade economy with living organisms.
The mycelium seeks out the microscopic root tips of massive living trees, pines, oaks, firs, willows. The fungus physically wraps itself around the root, creating a biological sheath.
That sheath acts as an interface. The fungal network acts as an expanded secondary root system, reaching crevices the tree roots could never access, scavenging phosphorus, drawing in deep water, funneling it up to the tree.
And in exchange, the tree shares its wealth. Through photosynthesis, it converts sunlight into liquid carbon sugars and pumps those sugars down into the fungal network, a mutually beneficial underground economy.
It's incredible. But here's the data point from the sources that truly stopped me. This ancient organism is actively invading and conquering the concrete jungle.
Yes. The Global Urban Evolution Project, or GLUE, tracks how wild species adapt to human cities, and they found Helvella lacunosa thriving right in the middle of urban parks.
And urban environments are brutal evolutionary testing grounds. You have the urban heat island effect. Concrete and asphalt absorb immense solar radiation, driving soil temperatures up to 8 degrees Celsius, hotter than a rural forest.
A massive thermal shock for an organism programmed to fruit in freezing winter rains. But the heat isn't even the hardest part. The concrete itself actively chemically alters the dirt.
Concrete is incredibly alkaline. Heavy rain washes over buildings and sidewalks, leaching highly basic compounds out of the cement and flushing them into the soil.
And the pH scale is everything in biology. Most forest fungi absolutely require slightly acidic soil to survive. Runoff spikes the pH, turning it highly alkaline. It's an environment that should instantly kill a delicate forest mushroom.
And yet, the Elfin Saddle persists. It shows phenomenal capacity for rapid evolutionary tolerance. Urban lineages, like Helvella huangi in Beijing, are adjusting their enzymatic processes to survive extreme heat and toxic pH and actively fruit in polluted urban spaces.
But even with its adaptable genetics and volatile rocket fuel, it is not invincible. Out in the damp forest, it faces a truly terrifying nemesis.
Yes, the specialized, deeply unsettling parasite known as Hypomyces cervinigenus.
The Hypomyces genus represents mycoparasites, fungi that exclusively hunt, infect, and consume other fungi. And this one has evolved a highly specific affinity for hijacking the tissue of the Elfin Saddle.
The infection mechanism reads like a sci-fi horror script. The Hypomyces releases microscopic asexual spores, illyriospores, and conidia. They land on the waxy surface of the Helvella, germinate, and physically drill into the tissue.
Injecting their aggressive mycelium directly into the host. The resulting physical transformation is a complete biological override.
The elegant matte black cap withers and is enveloped by a creeping, ghostly white powdery mass of parasitic mold. The parasite stops the Elfin Saddle from launching its own spores and uses the height of the mushroom stem to elevate its own white spores into the wind.
But this brings up a massive, glaring biological contradiction.
The chemical defense. We just spent all this time explaining that this mushroom is saturated with monomethylhydrazine, an explosive neurotoxic grenade designed to sterilize its environment. How on earth does a delicate powdery mold drill into a vat of rocket fuel and survive?
Is it immune? Does it neutralize the toxin? It's a phenomenal question highlighting the escalating nature of evolutionary arms races. The exact biochemical interaction is at the bleeding edge of mycological investigation right now.
So based on similar host-parasite dynamics, what's happening? Because it's probably not just magically immune. Hydrazine is fundamentally destructive.
Right. It's highly probable that over millions of years, the Hypomyces evolved specialized enzymatic countermeasures, its own arsenal of specific esterases or acyltransferases.
Enzymes acting like microscopic chemical scissors.
Exactly. Before the hydrazine molecule can bind to the parasite's vital proteins, these enzymes intercept the toxin, physically cleaving its chemical bonds and neutralizing its reactivity.
So it has evolved the exact biochemical hazmat suit required to wade through the rocket fuel and consume the host. Microscopic chemical warfare. And the foraging sources are incredibly clear on the practical warning here. Under no circumstances should you ever harvest an Elfin Saddle showing signs of this white powdery infection.
It's the absolute golden rule of wild foraging. You must identify the physiological health of the specific specimen.
Because even if you perfectly execute the 5-minute rolling boil and leach out all the rocket fuel, a body-snatched mushroom is profoundly dangerous. The hypomyces mold pumps the tissue full of its own complex, unstudied secondary metabolites. Eating it will trigger severe gastrointestinal distress completely independent of the gyromitrin. A diseased mushroom is inherently unpredictable.
Despite the profound weirdness, the hidden rocket fuel, and the gruesome parasites, human beings have refused to leave this organism alone. For centuries, diverse cultures have cherished this fungus.
Yes, the ethnomycology, how human societies have culturally and nutritionally integrated it. The cultural dimension is every bit as fractured as the genetic one. The research coming out of Mexico captivated me.
Mexico possesses an incredibly rich history of engaging with wild fungi, second only to China globally. Deep indigenous traditions integrating over 500 species of wild mushrooms.
The sources highlight the Tlohica Picachu culture and the Purihipicha people in the high volcanic central mountains of Mexico. To them, this organism isn't viewed through fear. It's a deeply revered nutritional resource.
The nomenclature is telling. Locally, they call it Saratangansi, translated to Spanish, orejas de ratón, or mouse ears.
Or orejitas, little ears. It's a gentle, affectionate phrasing for a chemically aggressive organism. It implies a long-standing, respectful relationship. In places like the Cofre de Perote region in Veracruz, traditional female mushroom hunters known as hongueras safeguard this intricate knowledge.
Their ecological literacy is staggering. They track subtle microseasons. They know when the temperature drops, how humidity shifts, and exactly which host trees the mouse ears will emerge under.
And beyond ecological tracking, their chemical mastery. Centuries before Western science invented gas chromatography to identify gyromitrin, these indigenous cultures had mapped the toxicity.
Through empirical trial and error, they pinpointed the exact thermal preparation, the boiling times, and water discarding methods required to neutralize the volatile hydrazine safely.
A profound testament to traditional ecological knowledge. They solved the biochemical puzzle using observation. But then you contrast that practical Mesoamerican view with the historical perspective of Western Europe, and the narrative completely fractures.
The European relationship with fungi has always been culturally turbulent. In the 19th century, Swedish mycologist Elias Magnus Fries grouped these mushrooms under the genus name Helvella.
The linguistic root of Elfin Saddle. The rural European folklore saw these strange, dark shapes emerging from rotting leaves, and the cultural imagination decided they were miniature saddles used by poor elfin creatures riding through the dark woods at night.
It sounds whimsical, but sociologically it reflects deep-seated apprehension. In Europe, especially Britain, there's a strong historical undercurrent of mycophobia, irrational fear of fungi. Associating the mushroom with elves and the supernatural, subtly categorized it as unnatural and slightly dangerous.
They mythologized it to keep it at a distance, but swing the cultural pendulum all the way to Asia and the dynamic flips again. Academic papers detailing Chinese markets state that regional Helvella species are aggressively sought after and universally considered a precious food.
The economic and culinary status in China commands a respect comparable to white truffles or fresh morels in Western hot cuisine. It's a massive commercial enterprise.
Which brings us full circle right back to the cryptic speciation we started with. Because the mushroom they're eating in China is not the same organism growing in Sweden or California.
Exactly. The genetic sequencing proved Chinese lineages are distinctly separate species, which raises an unresolved question. Are they a precious food simply because of a different cultural framework, or are they biochemically distinct?
It's highly probable that isolated evolution resulted in Chinese species with much lower, safer concentrations of gyromitrin, or vastly higher ratios of those beneficial polysaccharides.
The genetics dictate the chemistry, and the chemistry dictates the cultural value.
It's just profound to reflect on how a single visual disguise acts as a mirror for human society. To a scientist, it's a map of 49 hidden species. To a Hongera in Mexico, it's a mouse ear, a vital nutrient. To a 19th century English villager, an elf saddle, a creepy relic. And to a chef in Beijing, a highly lucrative, precious food.
It underscores our human tendency to project our specific cultural frameworks, fears, and economic needs onto the blank canvas of nature. We see what we are culturally trained to see.
And as our tools become more sophisticated, peering deep into the mutational rate of a gene, our understanding shatters old paradigms and forces us to reconstruct a much more complex reality. Which brings us to the end of this incredibly dense map. Let's synthesize the journey.
We started by confronting a comforting illusion, the cosmopolitan myth of a single species. We watched DNA sequencing shatter that, revealing at least 49 distinct, isolated lineages under one disguise.
We deconstructed its hollow, deeply fluted stipe, channeling Gothic architecture to survive winter storms while creating a microhabitat mansion for invertebrates.
We navigated the terrifying chemical warfare, how it synthesizes monomethylhydrazine space shuttle fuel to create a toxic halo, depleting mammalian GABA neurotransmitters and potentially contributing to neurodegenerative diseases like ALS.
But we balanced that darkness with its pharmaceutical potential. The same organism yields polysaccharides capable of actively reprogramming human liver genetics to burn fat and lower cholesterol, while acting as a UV-activated factory for vitamin D.
And finally, we traced its vast cultural footprint, from Mexican hongueras to European folklore and Asian commercial markets, a testament to the hidden depths of the natural world.
It is. But before we conclude, there's one final, really provocative concept we need to examine. We briefly touched on the global urban evolution project and how specific Elfin Saddle lineages are rapidly adapting to extreme urban heat islands and toxic concrete.
The era of the Anthropocene, the geological epoch defined by massive human impact. We are forcing nature into a corner.
As urbanization continues to exert intense evolutionary pressure, we have to ask a deeply unsettling question. How will the extreme chemical factories hidden inside Helvella lacunosa mutate in response?
We already know it possesses the metabolic flexibility to synthesize both volatile rocket propellant and complex cardiovascular medicines.
So what entirely novel, completely undiscovered chemical compounds might the urban Elfin saddles of tomorrow be forced to invent as they fight to adapt to heavy metals, synthetic pollutants, and artificial soils?
In our drive to pave the world, are we unwittingly forcing the evolution of a completely novel, unpredictable biochemical arsenal?
That is a staggering thought to leave on. The spy is still out there, actively changing its passports, rewriting its genetic code, learning entirely new biochemical tricks just to survive the harsh synthetic world we are building around it.
Truly incredible.
So the next time you are walking through a manicured urban park or hiking in the deep woods, and you happen to spot a strange, distorted piece of melted black wax pushing its way up through the soil, pause and look a little closer.
The biology beneath the surface is brilliantly, aggressively, and terrifyingly alive.
The visual disguise might be perfect, and you might not have a DNA sequencer in your pocket, but there is so much more happening right beneath your feet. Thank you so much for joining us on this deep dive.